Thank you very much. First of all, I would like to express my thanks for the opportunity to tell you something about our work, which is also focused on cancer treatment, but perhaps it will be somehow new point of view from most of you, I guess. So we are interested in daily rhythms and their role in the cancer progression and even more importantly in cancer treatment and determination of patient diagnosis. And at this talk, I would like to tell you something about daily profile in microRNA expression in the colon and what we were able to find out about their role in the colorectal cancer. Why we are interested in colorectal cancer? That's because incidence and mortality of this type of cancer is extremely high in our country. And I can you see my mouse when I am pointing? Okay, probably not. So in the middle of this yellow square, you can see Slovak Republic, and this dark blue shows that incidence of colorectal cancer is really higher in Slovak Republic compared to rest of the Europe and probably also rest of the most of the world. So that's also topic of our research. And we were trying to figure out how to improve treatment and we are finding and we are investigating ways how to do it without additional cost. And that's always a very critical issue in treatment of any disease. And now I would like to show you that it is possible to improve treatment of disease, and in this particular case of colon cancer or colorectal cancer. And it is just by organization or logistic of the treatment and proper diagnostic of patients. So at this slide, you can see how it happens that someone suffer for cancer. This is genetic model of tumorigenesis, which was published by Fieron and Vogelstein in 1990. And it shows that not only one, but several mutations are needed before someone develops cancer. And these are those genes that are most frequently mutated. And you can see cancer stage, which is associated with occurrence of these mutations and which pathway is associated or used by this particular gene, which is not functional anymore once there is mutation in this gene. So it looks like the genetic background of colorectal cancer or cancer is quite well defined, but it's not so because there are many combinations how the cancer can emerge. And in addition to these crucial genes, there are many other regulatory factors that influence cell cycle progression. And that's why genetic screening is not that useful as it is expected, probably. I suppose that even whole genome sequencing of human cells wouldn't be really couldn't really determine if someone gets cancer because these mutations can occur later during the life and in the very specific sample, not just in the lymphocytes. And therefore, the classical colonoscopy remains still the major or the most frequently used how to determine colorectal cancer. And most cases of colorectal cancer are sporadic, and there is only a few percentage of patients, up to 3%, which can be determined in early stage by genetic screening. In addition to those regulators of cell cycle, there are also other new regulators that can influence what happens in the cells and how it happens that the cell loses control under its processes and become cancer cell. So during the last year, Nobel Prize was awarded for discovery of functions of microRNAs. MicroRNAs are so-called small non-coding RNAs that do not code protein sequence, but they act by nucleus by this biochemical pathway. They are processed by DROSHA and its cofactor DGCR, and then they are processed by dicer in the cytoplasm. At the end, the microRNA binds to so-called RISC complex, and it executes one of these three actions. MicroRNA bound to RISC complex can cause degradation of messenger RNA or the adenylation of its poly-A tail that is followed by degradation of a messenger RNA, or it just block expression of microRNA. So in all three cases, action of RNA is inhibitory. So microRNAs are generally inhibitor of gene expression, and according to the sequence, they can target specific genes. And it was really shown that all these genes that were shown to be important in development of colorectal cancer are in some or another way influenced by microRNAs. And knowledge about microRNA achieved critical level, so there are already clinical trials that are investigating their possible use in cancer treatment, and there are two main strategies. Firstly, microRNAs can be used to silence mutated tumor suppressor, or it can be used to silence oncogene. But in most cases, they are just used for diagnosis because they represented what is usually called liquid biopsy. Because cancer tissue express different microRNAs compared to healthy tissue, and these microRNAs are released into the circulation in such a high concentration that it is possible to detect it in the blood, in the general bloodstream. Another strong, powerful regulator of cell cycle is the circadian system. Circadian system regulates and drives rhythms with period close to 24 hours. And it is present in the body of all mammals, including human. Circadian system influences many physiological functions, not only colorectal or not only. Under normal conditions, our biological rhythms are synchronized by external ID cycle, by master oscillator, which is localized in the suprachiasmatic nuclei. And suprachiasmatic nuclei via humoral and signals influence peripheral tissues. So this is a scheme of hierarchical organization and everything is working properly. LD cycle synchronizes suprachiasmatic nuclei, and suprachiasmatic nuclei synchronizes peripheral oscillators. However, in recent society, where people frequently work on shift work, when we have artificial light, many nighttime activities, food is intake anytime during the day, during the night. So there are many factors that are influencing circadian system. And as these peripheral oscillators differ in their receptoric capacity, they are differently responsive to external factors. And under these conditions, when there are many external factors that are influencing circadian system, these peripheral oscillators can be disconnected from the master oscillators, and there is general desynchronization of the human body, which usually causes worsening of existing diseases. And who in 2007 accepted circadian hormonal disruption as a probably carcinogenic to human, which was supported also by huge epidemiological studies. At the molecular basis, the circadian system executes its effect via so-called clock genes. They are expressed in the circadian in a rhythmic manner. There are two clock genes, PER and CRY. PER have 3 homologs, CRY have 2 homologs, and their expression is driven by clock and BMAL1 via regulatory region E-box. When protein products of PER and CRY achieve critical level in the cytoplasm, they create heterodimer, which is translocated back into the nucleus and inhibits their expression. So there is so-called feedback loop between expression and translation. There are also additional loops in the circadian oscillator. One is executed by nuclear receptors ROR and reverb alpha that regulates expression of BMAL1. ROR induces expression of BMAL, inhibits it, and they are exerting their effects in opposing time of the 24-hour cycle. That means that they are, in this way, they are inducing rhythmic profile and BMAL1 expression. Practical output from this knowledge is that it is important to consider at which time chronotherapy would be applied. These are results published by very famous group of Francis Levy, who showed that timing of chemotherapy is very important to achieve best possible effect and to minimize negative side effects. For example, very well-known oxaliplatin is best to be applied at 3 o'clock afternoon, and we know all that in most cases chemotherapy is applied usually at one time of day when nurse has time or when there is free time in some schedule. So this is our theory. Excuse me. So this is our theory that both circadian system and microRNAs influence the cancer progression, and we are investigating they affect and if they can interact. So firstly, we perform screening and of microRNAs expressed in colon tissue and reveal hundreds of rhythmically expressed microRNAs with acrophase or maximum levels showing predominantly at the beginning of night time. There is some cluster with maximum expression during the middle of the night time, and there are any or very low number of microRNAs expressed during the daytime. I must add that these samples were taken in rat, and the rat is active during the night, so this is inverted. The maximum of microRNAs expression is at the beginning of active phase in rat. In human, it would be opposite. Also, number of target genes and regulatory pathways is highest at the beginning of night time. At least those pathways and target genes that are regulated by microRNAs that shows acrophase at this time of day. This slide shows you which pathways are influenced by microRNAs with maximal expression during the daytime. Those are pathways involving p53, apoptosis, EGF signaling, angiogenesis, and inflammation. There is only one microRNA that influences p53 and shows maximum during the lifetime, but this effect is not specific. As you can see, it's shared with those microRNAs expressed during the nighttime. Also, biogenesis of microRNAs shows daily rhythm predominantly at the level of DGCR, which shows daily rhythmic expression in liver, heart, and kidney. We validated rhythmic profile of microRNA 150 by PCR after next generation sequencing and then investigated in functions. We associated its role with expression of two tumor suppressor, BCL2 and MIP, and measured the rhythmic profile in DLD1 cells. This is line of colorectal cell lines, and showed that expression of BCL2 and MIP is lowest at the time when expression of microRNA 150 is highest. This gene just shows that transfection by 150, microRNA 150 was effective, and also inhibition by inhibitor of microRNA 150 was effective. We were able to prove that administration of microRNA 150 was able to decrease metabolic activity of cancer cells and administration of inhibitor increased activity of cancer cells. Also, administration of microRNA 150 decreased expression of oncogene CRY1 and increased expression of tumor suppressors PER2 and CRY2. And also, we proved effect of microRNA 150 on individual cell ones, which were influenced by camptothecin, which is cytokine. At this slide, you can see fluorescent staining on caspase 3 and 7. This slide shows you control cells. At this slide, you can see what happens when we add camptothecin. So this is comparison between this column and this column. Of course, camptothecin increases apoptosis, but when we apply camptothecin microRNA 150, response to this cytostaticum is much stronger. Based on these results, we conclude that microRNA 150, which shows a rhythmic profile in colon, can be exert strong tumor suppressor effects in cancer, which are probably most effective at the time when expression of this microRNA is highest. That means at the beginning of nighttime. And most of these effects are executed by interaction and strengthening of signaling by pathway related to p53. And at these results, I would like to thank you very much for your attention.